Welcome to the Bioinfo Cloud. In this class, we'll discuss about a brief or we can say an overview of association mapping or GWAS by using the TASSEL software. Let's start the class. So for mapping or association study or any kind of mapping like linkage or association mapping, we require phenotype and the genotypic data. So here we have a genotypic data in the form of SNP marker. You could have in the form of SSR or any marker. So this is the data in the form of SNP marker. You can see this position is polymorphic, hence we named it as a single nucleotide polymorphism, and this is the phenotypic data. Now we have to connect our phenotypic data and genotypic data, and based on that we will able to identify a particular region, location, SNP or marker for a particular trait which we are going for analysis. Okay, so what are the steps in the association mapping? So for that you need a phenotypic data in the form of replication, or you can calculate a mean from that application. You can have a data from multiple traits and multiple environment or multiple year you can perform. That will that will be good, and it will also give us reliable result if we perform data in the multiple environment. Then we could have a genotypic data or marker data in the form of SSR, SNP, FLP, anything we can consider. Like then after we have to calculate linkage disequilibrium from this marker data. Then after we'll calculate population structure or relatedness. Then only we can perform the association study with by choosing a model like GLM, MLM, general linear model or mixed linear model. In general linear model we do not consider kinship matrix or population structure, but in mixed linear model we have to consider relatedness or the population structure. Then when we perform this, we will get a marker which is linked to a particular trait of interest. Then further you can identify genes within that LD region of that marker, or if you get good gene, then if you want to confirm that, then those gene you can go for the gene cloning or positional cloning. Then you can confirm it with the help of genetic engineering, CRISPR or something like that. You can knock out the gene and then you can confirm the function of that gene if you get any gene from this QTL mapping. So what is the flow chart, how we can perform? So we must have a genotypic data in the HapMap format. Then we'll do imputations to fill a missing value. Then we'll have to filter our data, that is the minor allele frequency we have to keep like 0.05. We have to calculate the LD, then kinship matrix, PBCA, population structure. Then we'll join the data and then we'll select the file and then we'll perform the study. So we'll see one by one how we can do it. Please download this TASSEL software from this website. Okay, here you can download based on your computer requirement, that is 64 or 32-bit. Then open your TASSEL software. It will look like this. Okay, now we have to check our data format. So this is our phenotypic data format. Okay, so this is our genotypes here. Okay, and these are our traits. This is a MAGE data. Okay, so these are our trait ERHT, DPOL. Okay, for the missing value you have to define like NaN, or you can also write like this NA. For the missing value you can write like that. So this is your phenotypic data. You can save it in the text format or in the notepad. Then for the genotypic data you should have in the HapMap format if you get from the GBS data. Okay, so this is the GBS data which is in HapMap format. In this HapMap format, these are the markers, these are the alleles and the markers and the positions, whether it is present in sense or antisense strand, plus denotes the antisense strand, and these are the like assembly and all. You can leave it like that only. And when you scroll them, these are the your genotypes. these are your genotype ID and your data in the form of diploid, or you can see CC means data are here in the form of diploid. You can also have your data in Unicode or the single nucleotide like this. Okay. Now we'll open our TASSEL software. We will save these data in the text file in a particular folder. Then we can open the data from this option. This is our genotypic data which is in HapMap format, and this is our phenotypic data. So we'll select all to both of the data at once by pressing the control, then we'll press open. Okay, so this is our phenotypic data already here. Okay, as we have seen in the TESSEL, Excel, and this is our genotypic data. You can see your genotypic data here. And how you can see this read this genotypic data here. So here you can see number of taxa or genotypes are here 281. And number of sites or SNP here is 3093, or we can say 3093 markers are there. Okay. You can also check the chromosome wise at which chromosome how many sites or SNPs are there. Okay. Now when you see this data, so this data is showing like in dual color, or three colors are there. So the blue one and this yellow one and the green one. Okay, so these are nothing but a major allele, minor allele, or the heterozygous. These some at some position you will get a heterozygous data, okay, or at some position you will get a N like nucleotide or missing data. You can also see like major allele from this option, minor allele, okay, or all kind of allele. Now what you have to do, we'll just see a overview. So we have to first, if your data is having a missing value more than 50 or 60%, 70%, then you have to go for the imputation to fill those missing value. So we'll run an algorithm. I'll explain this in detail in the later classes how we should impute. So you can impute from this option. Okay, this will fill your missing value. Okay, you can go to impute and then press LD imputation. When you press this OK, this missing value, this will run. If your data is too large, it will take like half an hour, but this is a very small data. Okay, so it just complete in a minute. Now you can see your data, the missing value is not there. You can also remove the heterozygous from your data, then go to the data and then select homozygous only. Now we have selected homozygous only. Now heterozygous are not there in data. Now the next step is we have to filter our data. There are some filter we have to put before we start analysis. So these are something like minor allele frequency, maximum allele frequency, heterozygous proportion. We can filter it out. But here I am telling you only the minor allele frequency, that is 0.05. This is the standard one. You can increase and reduce as per your data set. So we are putting here 0.05. So what is minor allele frequency? So it is saying that if your data is having less than 5% of the data which are present in different kind of are showing a different alleles, so that data may be due to the error, or because of some reason it will get just deleted from the your data. So we'll set a filter 0.05. Rest of the thing will leave as such and then press OK. So this is our data, we have filtered it out. Okay, now our genotypic data is ready. Okay, this is our phenotypic data. Now let's check the our flow chart. Our data, genotypic data is present in HapMap format. We have performed the imputation, we have also filtered it. Okay, and we'll make it like we have done this. Now we calculate the LD and the population structure. Okay, so what we do, select your this file which you have filtered, go to the analysis, go to the relatedness or diversity, go to diversity, then press linkage disequilibrium. You can have a full window, full matrix, but we are just making a sliding window. and window size you can also reduce or increase. I am just giving it 30. So this data is very small, so the result is completed. So this is our LD data table or result. How we can analyze this? We have to go to the result and then select LD plot. So we can select our LD. You can also just make it scroll up, scroll down. Okay. Now this site is your R square value. So R square is more than 5.5. We consider is a good kind of uh, SNP, or we can say LD is there, or we can also check the p value. Okay, you can see the scale from 0 to 1. I'll explain this what how we should consider and what is the significance of this LD map. Okay, so we have to check the LD before we perform analysis. Then we'll calculate the kinship matrix. Okay, then go to the analysis, select your file, go to the analysis, go to the relatedness, and then press kinship. When you press kinship, just press OK. This is your kinship matrix you got. Okay, also calculate your PCA. So for that, select your file, go to the analysis, go to the relatedness, and then select PCA from this option. You can make component or principal component like 3, 4, 5. I am keeping 5 as default. Then press OK. When you press OK, it will take a minute and then just complete it. You can see there are 5 clusters. Now our phenotype file, genotype file, kinship file, or the PCA file is ready. Now we can go for the analysis. I'll show you the slide so that you can get it. We have calculated the LD, PCA. Now we'll join our data. So we have done this. Now we'll join phenotype and genotypic data, and then we'll select all the file and then perform the study. Okay, so this is our all the file. So select the file, like first you will select phenotype file and then here genotype file. This is here in the sequence. Okay, press and select both the file by pressing the control. Okay, then select phenotype and genotype file. I have selected this here. Okay, this is our phenotype file, this is our genotype file. Then I'm going to the data and I'm joining them. So go to the intersect join. Okay, now you can see this is your genotype. You can see like this is your genotype or this is your phenotype. Okay, so we have joined the file. Now to perform association, here is the option association. We have to select the files. It requires some file like this, our joined file. So select this, then require our PCA file. This is our kinship file. So I have selected that file. We have joined this file. Then we require PC. Where is the PC? This is the PC. So we have selected all this file, kinship file, PCA file, and our joined phenotype and genotype file. You can see MDP traits means this phenotype and the genotype file. So before performing association, just select all these three files. Then go to the analysis and then go to the association. Now you have a model like GLM, MLM. So GLM model, you don't need the PCA kinship matrix. It's don't consider that. But we are doing MLM, mixed linear model. I'll tell you the importance of general linear model, mixed linear model, or some other advanced model like farm CPU, blink, bulb, by using the GAPIT in the later class. I'm just giving a brief of this TASSEL now. So I'm just selecting the MLM only. Okay, when you select MLM, this kind of window will appear and then just run it. Okay, so we can save our output. So tick here and then browse. When you browse and give a particular folder here, I'm making in like in desktop classes GWAS. So I'm making a folder here, result MLM. I have selected this folder and give the name like MLM. Save. You have one more option, you can filter it out based on the p-value, that is 10 to the power 3. So I just selected this. Okay. When you press OK, the analysis will start. If your data is too large, it will take like 15 minutes, 20 minutes, but this is small data, so it will get in a minute, complete in a minute. Now this is done. Okay. How you can check your data? Go to the file, open your result data. Okay. So result data, this is our result data. So I'm just okay. When you see your result data, where we have given the location, so there will be many like data set will see like fact, states, residual. So we have to open this file, this state one. Okay, so select this state file, then go to the result option and then select the Manhattan plot, because we see Manhattan plot to check significant SNP. So you can select a trait, different trait for Manhattan plot like year HT. So I'm showing. So this is your Manhattan plot, which we generally use to depict or visualize our SNP. Here you can see in the y-axis you will see the p-value, because you already know that p, on the basis of p-value we check our markers with the it is significant or not. So these are our markers, and the different color are showing a different chromosome like 1 to 10. So how we can interpret in TASSEL whatever LOD value, you might heard about that. So in TASSEL whatever comes after 10 to the power minus 3, so this is the cutoff. Okay, so whatever is coming after this, we'll consider it a significant SNP for this particular trait. So for year HT, if you want to interpret, so you will see the result at this SNP. Okay, when you press here, it will also appear like this is a chromosome 1 and this is the position. So likewise you can see the result. You can also check the result for other file. Go to the result, go to this, select this state and then see. You can also check for others. In this case we are getting two significant SNP. One is at chromosome 1 at this position and is another is chromosome 2 here. Now you have to also check your QQ plot. So QQ plot we just see. So this is also available in the form of different traits. So we are just checking for ERHT. So this is the window from which we can plot the trait. So we are selecting this, importing it from here, then OK. So this is for the ERHT. Now this is QQ plot as just tell us about the distribution of our p-value or the SNPs based on the p-value. So when this uh, data, or we can say the QQ plot shows us that these lines, which is SNPs, are following or exactly coming same as this line, it means our data is not we can say good or the normal. So it must deviate from this line which is expected. So this is our QQ plot. So whenever we show our result, we have to show in the form of Manhattan and QQ plot. So there are more advanced options are there that I will discuss it later. Okay, one thing you can do, you can also check your diversity by using the your phylogenetic tree or distance matrix. Okay, so suppose I am making the tree from our genotypic data. For that, select your genotype file, go to the analysis, go to the relatedness and then select your create tree. And they are the two methods you know already, neighbor joining and UPGMA. So I am selecting neighbor joining, press OK, this is your tree. But this you won't get able to interpret. So we have some other options in the result. Okay, go to the result file, select this type of tree, second option. it will take some second. Okay, this is your tree, you can select, but this is also not easy to understand because we have a 281 genotypes. So for that you can select the type and then select the circular. So you'll see this here in circular form here. And you can count the clade, means how many clades or clusters are there in your genotype, and you can also show the genotypic diversity. You can also like convert it. There's some other option like unrooted. This, from this you can easily count the clade. This is like 1 2 3. Likewise you can count the clade to just check the cluster or the subpopulation in your pop, uh, population or panel. Okay, so I'll show you one more thing in this. When you save the result from the result folder, okay, it will just appear like this. So this is our main file, the state one, where we have a SNPs. So we'll open this, just double click, it will open in notepad. Just copy it and then paste it in the Excel. So this is the result you just see. Okay, here you'll see many things. I'll just explain you one by one in a brief. Like this is your trait, okay, and this is your marker name, chromosome position, degree of freedom, FP value, additive effect, marker R square. But here just I'm explaining you these two things, which is good. So the genetic variance, residual and likelihood. So these are the result table. So we'll get a n number of SNPs are there. So we are interested in the lowest p value because you know the p value which is lowest is considered as a most significant one. So for that, this is our p value. We can easily sort it. Like select your p value, then go to the data and then sort it based on lowest to small. Expand the selection. Okay, now you can see the lowest one. So this is nothing but a 6.4 10 to the power minus 5, or you can say the point after point how many zeros are there, 2 4, and this is 5, so we are writing it as a 10 to the power minus 5. So this is the lowest one. So we'll see in our result we got a marker. This is the one, that this yellow one at chromosome 1 at this position. This is our physical position of this marker. At this base pair we found a marker at chromosome 1 which is found to be significant with this trait here HD. So what we'll write in the result, so we have a marker, this is the one for chromosome which is present in chromosome number 1 at this position for this trait is showing us the strongest association with the lowest p value, that is 6.4 10 to the power minus 5, or the r square value 0.05. What we can do, we can also convert this r square value in terms of percentage. So how we can do that? We just multiply it with 100. So this will be a good representation. So this is 5.1. So whatever value is coming after 5 or 4, we consider kind of a good association. So here we are getting these two. So in this result we only got these kind of two markers. So how we will represent this data? I'm just showing you. So this is our output, this is our trait dpol, and we got some SNPs here, more than 3, and this is our QQ plot. So we have to represent like this, and this is our table which we have processed in Excel. These are the traits, marker, chromosome position, and this. So now we can represent our data in this type. So I have just explained this in your very short and overview. In the detailed class, I'll just explain each and everything in very with my very minute detail, and the significance, how we can use and what is the significance of this. Okay, so this is all about TASSEL. So I am stopping here now. Thank you very much for listening.